Hello, everybody. Very good afternoon to all of you. And I
Good
hope
evening, sir.
enjoying the course. We will start in a while. I'm just waiting for some more people to join. So we can just wait for 2 minutes and then we can start. Again, let me introduce myself. I'm Gunjan Mehta. I am the course instructor for this course. And today I have my PhD student Akriti Kumari with me to help us with this demonstration. So while we are waiting for some more people to join, in case if you have any other question, concerns from the lectures or even from the previous demonstration, we can discuss it now. So feel free to ask questions. You can just unmute yourself and you can ask question.
Good evening, sir.
Good evening, Bhagat.
Yes, sir. Sir, actually, I've joined the course very recently and I've missed out the previous sessions of the question and answer or whatever. So, can I get the recording of it, which would help me?
Yeah, so see, all my lectures, video recordings are already available. And
Yes,
we have just completed one live demonstration previous week, the same day, same time on last Friday.
yes, sir.
So
I've
that
missed
was,
it.
yeah, that is also available on YouTube.
Okay, yes, sir.
So,
But
uh,
how do I get the channel? What name it is?
okay. Vignesh, can you please help them getting the YouTube link for the previous class, previous
Yes, so
demonstration?
we have the group in the mail, so can it be shared so that it's available
Sure,
for everyone?
sure. So after our demonstration today, I will share that link. Okay,
Yes,
in
yes,
the
sir.
group.
Thank you, sir.
Okay. Any other question? We have 19 participants, and okay. So let us start then. Um, so today. I will show you how to do the 5D imaging of a yeast cell. Okay, so I am sitting in front of a fluorescence microscope. So this is the fluorescence microscope that I showed you, and I explained all the components and how it works, how the light path works in the last demonstration. Okay, so this is the same fluorescence microscope that we will use today to capture a 4D and 5D image of live cells. Okay, so this is the microscope, and this is the PC connected to the microscope. So this is fully automated microscope. So all its components can be regulated using this software. Okay. So what we are going to see today, you can see here, our samples are Baker's yeast cell, okay, which are known as Saccharomyces cerevisiae. Okay. So this is the yeast that we generally use for making the breads. Okay. So the same yeast, we have several proteins fluorescently labeled in this yeast. Okay. So here you can see, first we will image this sample in which there are three proteins which are labeled with three different fluorescent colors. So ACE1 is one of the transcription factor, which is GFP tagged. SPC42 is the spindle pole body protein. So it is labeled with BFP. BFP stands for blue fluorescent protein. NOP1 is a nucleolar marker. It is a nucleolar protein, and it is also tagged with BFP. So see, there are two proteins tagged with blue fluorescent protein, means blue color. Okay, but their localizations are different. Okay, so even they are labeled with the same color, we can see them separately in the cell. And MLP1 is the nuclear membrane protein, which is labeled with M cherry. So we will take a Z-stack image in three colors: GFP, blue color, and M cherry. Okay, to see the co-localization or localization of these three proteins in the live yeast cells. Okay, so feel free to ask questions. If you have any question at any point, just unmute yourself and start asking the question. Okay. So now I will show you how I am making the sample. So for sample preparation, I am taking this cover slip. It's a very thin cover slip. Okay. So generally, we will image the cells with 63x oil immersion objective lens, and for that it is always better to have a very thin cover slip. Generally, if you take the glass slide, which is of little more thickness, you cannot focus the cell from the bottom of the glass slide. Okay, so it's better to take this kind of a thin cover glass for better imaging. Okay. So this is the cover slip that I have taken, and now I'll put drop of yeast cells on this. So yeah, after you can handle the. So this is my sample in which the yeast cells are pelleted. So you can see the pellets here. Okay, so this is a pellet of freshly grown yeast cells, and I will take just 3 microliter of this sample on the cover slip. So I have taken 3 microliter of this sample, and I'm putting it on the cover slip. Now, ideally for imaging, I can just put another small cover slip on top of it, and I can take it for imaging to the microscope. Okay, but I will not do that because I want to do live cell imaging, that too for little long time. Long time means at least 5 to 10 minutes, right? So if I just put the another cover slip and take it for imaging, then this water will dry, and eventually you will see the shrinkage of the cells and those things. Okay, because the water will be evaporated. So to avoid that thing, I have made this kind of an agarose pads. So here you can see, so whatever the media you grow the cells, let's say for yeast, we use the CSM media for culturing. So in the same media, we have added 2% agarose to solidify it. And I am taking a small piece of this media like this, okay, and I will put it on top of these cells. So this is known as agarose pad, and I will put it like this. And I will gently trace it from top so that my cells will be evenly distributed. They will be monolayered and evenly distributed under the agarose pad. And because of the agarose pad, which has all the nutrients and all, I can do the live cell imaging for even long time. Okay. So now I'll take this cover slip onto the microscope. And as you can see, currently the 63x oil immersion objective lens is in place. So this is the 63x oil immersion objective lens. So first I put a drop of oil on my sample. So see, I just put the drop of oil here. And I'm sure by now you must be clear that why we are putting the oil to capture the more diffracted light into the objective for a better resolution. Okay, the oil immersion objective lens are used to improve the light gathering capacity of the objective, and it can collect more diffracted light, and that helps in improving the resolution. Okay, so I kept the sample. Okay, so see how my sample looks on the stage. So this is the objective lens. On top of that, I have kept my sample. You can see a thin cover slip, and on top of that, we have kept an agarose pad. Okay, and now I'll close this chamber to avoid the exposure of stray light. Okay, okay, and now my first job is to see the cells, focus the cells by looking the cells into this eyepiece under the transmitted light mode. So first, from the software, I will switch on the transmitted light mode. And I will focus my cells here. So now I'm focusing my cells. To see my cells, and now I'm finding the best field where I can see enough cell density for imaging. Finding a good field for imaging. Yes. Okay. So I could focus the cell here. I can see the cells there in the iris, and now I will show you on the computer screen that how they look. So here, can you see these cells? And I can see that they are not perfectly focused. It seems like little uneven illumination. So I'm just Okay, so now it's perfectly focused. Okay, so see, now the cells are perfectly focused here. Okay, and these are the yeast cells. Some of the cells are dividing, that you can see like this, and some of the cells are in the G1 phase. Okay, so you can see that cells are at different stages of their cell cycle. So this we are visualizing the cell morphology using the transmitted light. Okay, this is generally any school microscope can show you this kind of structures. But now using the fluorescence microscope, I will show the internal details. Like as I mentioned that we have labeled three proteins, actually four proteins with fluorescent labels, one protein with GFP, one protein with M-cherry, and one protein with blue fluorescent protein. So now I will set the exposure time and the light intensity for each of this color. Okay, so what you can see from here, I have to adjust the light intensity. So first, let me adjust the exposure time for green fluorescent protein. Okay, so I will go to the green channel, and here you can see what I am seeing here. Now, whatever the bright cells you are seeing, those are the dead cells. So don't get happy by looking at those just 3 or 4 bright cells which are there on the field. But this is the real fluorescence that we are looking for. You can see the nuclear fluorescence. Now see, here you can see the nuclear fluorescence. On top of that, you can see the entire cell also, right? So you are seeing the entire cell because of the autofluorescence. So yeast cells show this kind of autofluorescence in green channel. So you are seeing the entire cell because of the green, because of the autofluorescence. But within the green cell, you can see this kind of a nuclear fluorescence, as you can see clearly in this cell. The nucleus is super green, right, compared to the rest of the cell. In all the cells, you can see there is some nuclear fluorescence, and entire cell is also green because of the autofluorescence. So for this GFP imaging, see, I have kept my light intensity to 20%, which I will slightly increase to 30%. Okay, and the exposure time is 465 milliseconds, which I will adjust to just 100 milliseconds. Okay, so here is the sample, and you can see now exposure time for green channel is adjusted. Now I'll move on to the next channel, that is blue channel. So I'll select this blue channel, so you can see the automatically the appropriate filter is selected. So this is the filter for blue color, and this is the LED for blue wavelength, that is 390 nanometer LED. So how is the blue signal here? You see, so you can see there are two proteins tagged in blue color. One is the NOP1 protein, which shows the nucleolar. It's a nucleolar protein, and you are seeing that in blue color. The rest of the blue color is because of the autofluorescence in yeast cells. And what are these bright cells you are seeing? Those are the dead cells. Okay, so we should not focus on that. But in the rest of the cells, you can see blue fluorescence, right? And for blue fluorescence, I think this exposure, I am setting the exposure time to 100 milliseconds in the software. And the light intensity is 20%, or maybe I can increase it a bit to 30%. Okay, I increase the light intensity to 30%. And now I am switching to the red color to see the MLP1 M-cherry protein. And here you can see. So one thing you can observe that the red color dead cells, so the dead cells are visible in all 3 colors. Okay, because that is not the real fluorescence. This is M-cherry, and so now you can see the red fluorescence. I will adjust the light intensity to 30% or 31%, 30%. And that is it. This is M-cherry, so 575 is the LED, and this is the filter for M-cherry that I am using. And with this, you can see the nuclear envelope in the cells. Okay, now what we'll do, I adjusted the exposure time and light intensity for 3 color imaging for all these 3 channels. And along with that, it will take the image in bright field also. Okay, so there are 4 channels set up here: blue, green, red, and gray. Gray is for the bright field imaging. Okay, and now we have to define the Z positions. Okay, because we want to take the Z stack. So we will define that. So how to define the Z position? So in the software module, you see, you have to select what you want to image. Whether you want to go for Z stack imaging, you want to go for time lapse imaging. If you click on this time, then software will show you some options for setting the time interval, that at what time interval you want to take images. If you select the Z in the software, it will show you the options to set the Z position, that what should be the step size, how many Z steps you want to collect. Okay, so now I'll show you how to collect the Z step, means how to define the top and bottom position for the image. So see, here software is asking me to define the begin position and the end position. So what I will do, I will just move the stage up and down in Z to see where the signal, where the cell starts and where the cell ends. Okay, so yeah, you can hold the camera here. So see, currently it seems like I'm at the center position. So I will, using the uh, Z position control, I am going to one end of the cell. So you can see that the cells will become defocused. Okay, and I will define this position as the start position. And now I will go to the other end of the cell and define this position at the end position. Okay, so then software will show me like this. So see, I define the begin and end position. Okay, and you can see software, the begin position is this 1120 micrometer, the end position is 1113 micrometer, and this total Z size that I capture will be 7.29 micrometer. Okay, so generally the yeast cells are 5 to 6 micrometer in diameter, so it is okay. Like we need to collect 7.29 micrometer entire Z stack to capture all the information. And as here you can see how I'm defining the Z stack. So here I have clicked the option System Optimize. System Optimize means this software will automatically calculate the Nyquist, as per the Nyquist equation, that how much should be the Z step size. So as per the Nyquist equation, the Z step size it is showing is 0.27 micrometer. Okay, so at every 270 nanometer, it will take Z steps, and it will take 28 number of steps, that is 28. So it will take 28 Z steps to cover the 7.29 micrometer volume. Okay, so see, these are 3 parameters very important. I have kept the system optimized Z step size. So that means the software will automatically calculate based on the Nyquist equation that what should be the Z step size. So the recommended Z step size is 0.27 micrometer. And with this step size, software will collect 28 Z sections, and it will cover the entire volume of 7.29 micrometer. Okay, so we are all set for Z stack imaging. Okay, I have set the Z position, and there is also an option to add the time. As I showed, if I click on this time, then software will ask me whether you want to take the time-lapse movies. So if I take the image now, it will just capture one Z stack image in all 3 colors. But if I set up the time that, okay, I want to collect this image at every 5 minutes, then microscope will automatically take another movie after 5 minutes until I ask to stop. Okay. So let us first capture the time-lapse movie. Okay. Now see, while we were discussing all this, I was in the live mode, and while my cells were exposed to the fluorescent light, and that's why you can see the signal is bleached. Okay, so now there is no point of taking the image of this position. Okay, so what I will do to avoid this situation, I will just move the stage in X and Y and find out another field. So see, when I will go to the next field, you will see the better, you will see a better fluorescence. because this is bleached one. And now let us move to the left, and then you may find little bright fluorescence because here the cells are not bleached. Okay. Okay. So this is a new field, and I'll just stop here. So yeah, and now I will just press the start button. Here you can see a start button in the software. Okay, so if I press the start button, then what software will do? It will first take this 28 Z positions for the blue channel, then 28 Z positions for the green channel, then 28 positions for the red channel, and then 28 positions for the in the bright field. Okay, so let us start and see how it captures the data. So I'm just pressing the start button. So blue channel is captured. Now green channel is captured. Red channel is getting captured now. And the bright field image is being captured now. Okay, so this is done. It captured one image of 28 into 4 channels. So it collected so many images. Okay. And now I'll show you how it looks like. So here is the Z step. So you can see the cells will start from out of focus position. So see, this is the bottom position of the cells. Okay. And now I'll move through the Z position. So you can see the cells will come in focus. So this is the center position where the cells are perfectly in focus. And now I'm going top of the cell. So this is the top position of the cell. Okay. I'm just moving in the Z position. Now I'm switching off this bright field. So you can see the red fluorescence. Okay. So in red fluorescence also, in Z you can see the cells are completely defocused at the bottom. Then I took all the Z section, I came to the middle of the plane. So you can see the proper fluorescence signal. And again, I'm going to the top of the cell. Okay. Similarly, the green channel, here you can see. And similarly for the blue channel. You can see the specific nucleolar localization of that NOP1 BFP protein. Okay. And now what I want to do, I will just merge all these 28 Z positions. So there is an option in the software which is known as maximum projection. Okay. So if I click this button, then it will generate the maximum intensity projection image. And you can see this is the merge of all 28 Z positions. For blue color, for green color, for red color, and in red color you can see, I'll just reduce the contrast a little bit. Okay. So can you see the nuclear membrane is labeled with MGel. So you should see the nuclear membrane. Okay. And if I turn on the cells and I will reduce the brightness of this image. Okay, and now we will merge these channels. So I switch on the red channel, green channel, I'm just adjusting the brightness and contrast of each of the channel. So we can see a nice 3-color image. Okay. So see, here you can see in these cells, these are the nuclear pore complex protein and cherry. The blue color thing that you are seeing is the nucleolar protein, and also very faint green color fluorescence you can see here. Green color protein hasn't expressed well, so it is not really giving a good green fluorescence though. But if it is properly expressed, then you could have seen a proper green color signal. Okay, so basically this is how to make the 4D or 5D or 6D images. Okay, so see in this image, there are 3 colors and XYZ positions are recorded, right? So this is known as the 4D image. Okay, but if I add time to this image, let's say the software automatically captures image at every 5 minutes, then it will be a 5D image because the dimension, one more dimension is added, that is time. Okay, and here I captured only this field, okay, but there are some more cells on the left, there are some more cells on the right. So I can also define these positions. I can ask the software that first you capture this image or this position, then you move on to this position and capture another field here, and then you move on to this position, capture another field there. Okay, and then stitch the image. So you can see all the cells from this position to that position. So that is known as image stitching and multi-position imaging. So that also you can define to the software for generating 5D and 6D imaging data. Okay, so that's all about the 5D or 6D imaging. If you have any questions, feel free to ask now. Any question?
Sir, I have a doubt, sir. During when you like change the colors, you only you sometimes change the blue color, like the multiple colors were there, right? For red, you change the yellow color and the purple color. But why did you change that opposite colors?
So while acquisition or while processing?
No, I don't know like how to say it, but when you got the image
Okay.
at that time, you changed the different colors like 30%, 20%, that one.
You are talking about this one?
Yes, yes.
Okay. So see, there are 8 color LEDs in this microscope. Okay, and these are those 8 color LEDs you are seeing, starting from 390 nanometer to 440, 475, 510, 555, 575, 685, 747. So these are the 8 different color LEDs are there, and you have to set the power of those LED. So see, for most of the imaging, I have used roughly 30 to 50% LEDs. Okay. So as long as you can see your signal, you should not use LED to 100%, because if you use the LED 100%, it will unnecessarily cause photo bleaching and photo damage, right? So the concept here is that if you can see your signal with, let's say, 5% LED power, you should not go for 10%.
Okay.
Okay. So the light source, basically we are adjusting the intensity of light source using this thing.
Okay.
And the below here, what you are seeing, those are the fluorescent filters that we are selecting. So let's say for GFP imaging, I'm selecting this channel. So see, automatically that GFP filter is active. Now you can see the GFP filter is active. Okay. When I selected this green color imaging, and with that, only the 475 nanometer LED will work. With this filter, no other LED can work. Okay. So this 475 LED, which is set as 30%, it will emit the light in blue color so that you can see the GFP fluorescence. Does it make sense?
Sir, you should like green color one is only emitted, no, sir. But why is that one is like in very less percentage?
No. So see, for GFP, the light color should be blue. The
Okay.
excitation light will be blue in color, Okay, that is generally 488 or 475 nanometer light. It is blue in color, but it will the emission will be green.
Okay. But you can only change that yellow color only, no, sir. You can't change that green color, right? The 575 only you can change, not 510.
No, so see, currently the filter is GFP filter is selected, right? Here the GFP filter is selected. So
Yeah.
software will automatically highlight that with this filter only this LED can work. See,
Okay.
if I want to switch on this 575 LED, I cannot. Software will not allow me to switch on this LED because that wavelength is not compatible with this filter.
Oh, okay, okay.
So see, for GFP filter, the excitation wavelength you can see 450 to 490, right? And here only the 475 comes in that range, right? So that's why only one LED is active.
Okay, okay. Thank you.
Okay.
Hi.
Any other question?
Yeah, sir. Good evening, sir.
Good evening.
Sir, so when we capture the image, we don't have to do multiple captures. Like what I saw is with one image, you were just changing the filters and we could see the GFP, the M cherry and all. We don't have to do multiple captures. That is one thing, right, sir?
So see, there are two things. What initially I did, when I'm, let's say, putting any sample for the first time, I have to adjust the exposure time and light intensity for each of the color, right? So what I did, first I put the slide on the stage, and then
Right,
I
sir.
focus the cell using the bright field,
Mm-hmm.
and then for each of the color, okay, individually, I adjusted the light intensity and the
Right, sir.
exposure time here, which is 100 milliseconds.
Okay. Exposure time also needs to be adjusted for each channel, sir.
Right. For each channel, the
Okay.
exposure time may be different or may be same, depending on how much is your expression level of that protein. Okay. Let's say if one protein is is expressed in very low amount, then you need to go for higher exposure and even the higher LED power.
Sir, is this similar to what, like I have worked a little with FACS, so over there also, I think to work on the bleed through, we make sure that one channel is not passing to the other. So we minimize the strength of one of the colors.
Right.
Just
Exactly.
to.
This
Okay.
is the same thing.
Okay.
So
Okay.
let's say if I start using all these LEDs with 100% power, right? And if I
Right,
put
sir.
a very high exposure, then it may possibly create some bleed through in the adjacent channels, right? So that's
Right,
why
sir. Right, sir.
the concept is that you try to minimize your LED power as well as the exposure time.
Right,
Okay.
sir. Right,
If you
sir.
can see your signal in 100 millisecond exposure, you will not go for 400 millisecond exposure.
Right, sir. Right, sir. Right, sir. Yeah. Thank you so much, sir. Yes, sir.
Okay.
Ji.
Any other question?
Good evening, sir.
Yes, good evening.
Sir, I have a doubt regarding this Z stacking.
Okay.
So in that, we will adjust the parameters of the number of steps and the distance between the each steps we have to calculate, no, sir.
Right.
The distance we'll calculate using this software, the website of this Nyquist theorem.
Right.
How to calculate the number of steps, sir, for a given structure? For example, a E cell is like 4 microns in diameter. So how to calculate the number of steps? How many images have to be taken?
Okay. So see, just I'm giving you the example of each cell. Okay, because we just imaged that. So the theoretical size of the each cell is 5 to 6 micrometer. Okay. And if I have to capture the entire volume, then it is always better to go for little more, like 7 micrometer or 8 micrometer, so that the entire cell will be completely covered. Now,
yes,
you just have to define the start and top and bottom position. So what I did in the live cell, in the live imaging mode, while looking at the cells, I went to the bottom of the cell. Okay, so see, now the cells are completely in focus, right? But if I go to the bottom of the cell, then the signal will be diffuse. Okay, and I cannot see all these structures and signal and anything, right? So that is considered as the bottom of the cell. Okay, let me just do that. So suppose so see, now I am going to the live mode, so you can see the cells in live. Okay, And now this is the center position of the sample. Okay, but now I will go. Yeah, close it. Now I'll go to the bottom of the cell. You will see that the fluorescence is gone, right? Signal is gone.
Yes.
So I will define this position as a bottom position. And again, I will go through the top position. So again, the cells are in focus. See, now the cells will come in focus again. And now I'm going to the top position. So again, this is a top position.
Yes.
Right. And I will define this as a top position to the software. See, software is asking me both the options, that define your begin position and define the end position. So just by clicking this button, I can just define the start position and end position. So it will calculate that this is a distance of 7.29 micrometer that I have traveled.
Okay.
Okay. And then
Okay.
when I'm selecting the option of the system optimized, that means as per the Nyquist calculation, because the software knows that which objective I am using, which mode of imaging I am using, what is the refractive index of the oil, right? So based on all these parameters, it will calculate the Nyquist sampling distance, and that it is showing here, 0.27. Okay, that is 270 nanometer is the Nyquist sampling distance in Z. So it will take the Z steps at every 0.270 micrometer and total 28 positions.
Yes.
Make sense?
Yes, sir. Yes, sir.
Okay.
Sir, and what is this name of software, sir?
This is the standard software that generally comes with a Leica microscope. So this name of the software is LASX, LASX.
Okay, sir.
Okay, but this is a standard software that generally comes with the Leica microscope.
Okay, sir. Thank you, sir.
Sir, I have a question. Sir, why does the bottom and the top one looks blurred, sir? Like, what is the reason?
Okay, so see, when you are in perfect focus at the center of the cell, you can see all the structures and what are the fluorescent molecules you want to see, right? But if you go on top, those things will be defocused.
So, like, the light will generally pass through straight to the sample, no, sir? Like I don't understand that part.
Right. See, light pass straight, right? That is true. But your objective cannot focus the entire volume at one go.
Okay, okay.
Right. So your objective will focus only a small thin section of the cell.
Okay, okay.
So when you will go up, you will stop seeing all the fluorescent signal.
Okay, okay.
Similarly, if you go down, you will stop seeing the samples.
So is it like based on the strength of the sample, like we'll manipulate like which one is the top, which one is the bottom?
No. See, in the live mode, actually, we can see that, right? See, currently, this is a live mode, okay? And if I come to the center, then you can see this nuclear membrane labeled with that M-cherry protein. So you can see some signal, right? Because this is the center of the sample. But if I just move my stage up or down in Z position, then you will see that now the cells are defocused.
Okay.
Because I went up. Again, I am at the middle position. Now I go down. So this is again defocused.
Okay.
So that is how you have to define the start and stop position to the software.
Okay, sir. Thank you, sir.
Sir, I have a question.
Yes.
Sir, there was a lot of background signals. So is there a way by which, by adjusting the intensity, we can reduce the background, or is it once the video has been captured, then later you are may increase the contrast?
Right. So yeah, so see, background is this like this. See, now you are not seeing background, right? But suppose if I just push these values, then you can see you will start seeing the background. So that is just a matter of post-processing. So once the image is captured, you can do this kind of post-processing. You can adjust your brightness and contrast of the image to reduce the background.
Sir, I have another question. So when you prepare the sample, you put a slab of agarose on top of it.
Right.
So, this one we can use it not only for yeast but also for bacterial pellets.
Right.
Yes.
For bacteria also you can use this thing.
And another thing is for animal suspension cultures, can we use it? Blood cells?
Okay, for imaging animal cells, this cannot work. Okay, because your
Yeah,
you are talking
CO2 is
about
required. CO2 is required.
right. You are
Yeah.
talking about the suspension culture, right?
Yeah,
So
yeah.
this approach will not work. But in
Huh.
that case, there are some imaging chambers available. Okay,
Yeah.
maybe I can show you here. I have one most likely here only. Okay, so see, I have this imaging chamber here.
Yeah, yeah.
This is for cells.
Oh,
Okay, so it has 4 chambers. Okay, and it has a cover slip thickness bottom.
okay. Yeah.
So you have to grow your cells in this chamber itself. If you want to give 4 different treatments, you can give 4 different treatment, and then you can close the chamber. You can put it on the slide on the stage for imaging.
Okay. Okay, sir. So for adherent cells, that is fine. But for suspension cultures, you do have you centrifuge and bring it down, or
Right. So yeah, for suspension culture, you need to centrifuge down, and you need to use the same kind of a chamber for suspension culture. But there are some ways to fix their positions. Like there
okay.
are sometimes the collagen or those kind of substrates are used so that your
Okay.
suspension cells will not keep floating.
Okay. Okay, sir.
They will adhere to the bottom of the chamber.
Same. Okay. Thank you, sir.
Okay. Yeah. Any other question? Any question on the chat box? No, right? Okay, so if there's no other question, then we can end the demonstration now. And one more demonstration is planned for the next Wednesday from 4 to 6 p.m. And in that demonstration, we will show you another high-end microscope that is from Nikon, and that is a confocal and super resolution microscope. So we will show you how to do imaging in the confocal and with super resolution. Okay. Okay, so feel free to ask questions. You can just send emails or there's a discussion group. Just keep asking questions if you have any doubts. And also during the live sessions, also feel free to ask questions. Okay. Okay, then see you on next Wednesday. Bye.
Thank you, sir.
Thank you,
Vignesh,
sir.
can I leave?
Thank you, sir.
Welcome. Vignesh?
Thank you, sir.
Thank you, sir.
Okay, so let us leave the call.